Hello everyone. As all of us know, the double helical structure of DNA was given by two scientists, Watson and Crick. They also proposed a scheme as to how exactly the DNA replicates before every cell division. DNA being the genetic material, it's extremely important that it replicates. And of course, the progeny cells should have the same genetic information as the parent cell. Don't you think? Let's see what Watson and Crick suggested. They said that the DNA double helix will unwind and each strand will act as a template for the synthesis of a new complementary strand. The result would be the production of two progeny DNA double helices, each consisting of an old strand and a new strand. This model of DNA replication is called the semi-conservative mode of replication. But this model of DNA replication remained untested for several years following the publication of the DNA structure. This semi-conservative model can be distinguished from a conservative model of replication, in which the two parental strands somehow come back together after the DNA replication. That is, the parental molecule is conserved. A third model called the dispersive model also suggested that all the four strands of the DNA following replication will have a mixture of old and new strands. Finally, in 1958, Meselson and Franklin Stahl devised a very clever experiment which supported the semi-conservative model of DNA replication as predicted by Watson and Crick. Remember these points to understand the experiment better. E. coli takes 20 minutes approximately to perform one cycle of DNA replication. N15 is a heavier isotope of nitrogen. Remember, it's not radioactive, it's just heavy. Centrifugation means rotating something at a very high speed. This was done to separate the DNA content in a cesium chloride density gradient so that the molecules with heavier mass like N15, since it's heavy, would sediment down faster. Meselson and Stahl cultured E. coli for several generations in a medium containing nucleotide precursors labeled with a heavy isotope of nitrogen, N15. They took one sample, broke down the bacterial cell, extracted the DNA from the bacteria in the sample, and then centrifuged the DNA sample to separate according to their densities. They then transferred the bacteria to a medium with only N14, the lighter isotope. They took another sample 20 minutes later, broke down the bacterial cell, extracted the DNA from the bacteria in the sample, and then centrifuged the DNA sample to separate according to their densities. They saw the isolated DNA had a density that was exactly intermediate between N14 and N15 DNA. The result eliminated the conservative model. After 40 minutes, when the DNA has replicated twice, in generation 2, half the DNA was of the intermediate density and half was of the density of N14 DNA, a result that nullified the dispersive model and supported the semi-conservative model. Thus, the observation was consistent to the semi-conservative replication model, where each replicated molecule after one generation consisted of one new N14 strand and one old N15 strand. And that just about covers this topic of experimental proof for DNA replication. Thank you so much. For more of such videos, to do a quick recap of concepts, do stay tuned in this channel, Biology for Life.